Hi everybody, this is Sam Rhine, giving you a little overview of what we will be talking about this year in the Genetic Update conferences as we travel around the country. Just a little preview. We're going to break things this year into two parts. First, focusing on genes and cancer. Take a look at oncogenes, tumor suppressors, driver mutations, passenger mutations. Also take a look at cancer stem cells. Then we'll take a look at tissue engineering and the process of actually making human body parts, tissues, and organs in a laboratory. So for the overview, let me just show you a few pictures and see if you can see how they tie into these subjects. This gentleman has something very unique. I think you can see if you look very carefully right there. He lost part of his nose because of surgery that removed a tumor from his nose, and the new part of his nose in the circle was made in a laboratory. That's a man-made human nose repair. Now we're taking a look at chromosomes at random. That's a purple chromosome, orange chromosome on the left. See in the middle, if they break and exchange parts, then we end up on the right with what's called chromosome translocations, and all cancers caused by DNA damage, and chromosome translocations is the top number one type of DNA damage that predisposes to leukemias and lymphomas. So when it comes to chromosome damage that causes cancer, many times it is initiated with a chromosome translocation. This is HSE. That's a fancy word that means human skin equivalent, man-made skin we can make in the laboratory today, and that can be used for people, for instance, who've had severe burns to replace the skin they've lost as part of the therapy and healing for burns. This is, of course, the mitotic spindle. You all know and love the spindle, the centrioles, the spindle fibers, the chromosomes. You see we're in the middle of metaphase here, metaphase of mitosis. We'll spend a lot of time this year making sure everybody's square on the significance of mitosis and cancer. You all know, of course, cancer can form by too much mitosis, forming an excess of cells, and that, of course, would be a tumor. But we're going to look at mitosis as symmetric mitosis and asymmetric mitosis and see how that process fits into cancer formation also. So we'll take a good look at details of mitosis. Now we're back in the laboratory, and this structure that looks like a balloon in a petri dish is a man-made human urinary bladder. Sometimes people have urinary bladder cancer. We'll soon be able to make them a new bladder in the laboratory. Other people born with bladder abnormalities, and they can be fixed. And by the way, the cells they use to make the new bladder are biopsied from the existing bladder, so no stem cells used. We're using the adult autologous somatic cells to make the new bladder. These people are just to remind us that the number one cause of cancer death in the world is adenocarcinoma of the lung, and they're demonstrating the number one cause of lung cancer, and of course everybody knows what that is. Now we're back in a petri dish in a laboratory. You probably can guess what we're making here. That's a replacement finger. Those are the finger bones being made in the lab to replace digits for somebody who's lost a finger from some sort of an injury or complication of some sort. Here's another cause of cancer, one we run into every day. Ultraviolet light from the sun causes unique mutations in the DNA of the cells of your skin. That can lead to melanoma, basal cell carcinoma, squamous cell carcinoma. So those particular cancers, the cancer-causing agent, the carcinogen, is ultraviolet light. Back in the laboratory now, within the confines of that white plastic ring in the petri dish is growing a thin membrane which can be used to replace the membrane on the very front of your eye. That is a corneal epithelium we can now make in the lab. It takes about 2 weeks. Now, we mentioned mitosis a while ago, but keep in mind there's another process by which cancer occurs, and it's not too much mitosis, it's not enough apoptosis. And this is a picture of a cell going through the apoptotic process. Those cells, that process can be defined as cell death, programmed cell death, where mitosis is programmed cell birth. When a cell dies through apoptosis, it shrinks away from its neighboring cells. It goes through a process called blebbing. You're looking at the blebs on the outside surface of the cell. The nucleus fragments, you can see the nuclear fragments in the blebs, and then they break down into little tiny pieces called apoptotic bodies, which then are consumed by macrophages and destroyed. So we'll take a hard look also at how apoptosis affects tumor formation, and keep in mind, when it comes to chemotherapy and radiation for killing cancer cells, the number one cancer cell killing mechanism is also apoptosis. Now we're in the laboratory again, and this is a man-made mouse liver, and the foundation of the liver was laid down not with an inkjet printer, but with a liver cell hepatocyte jet printer to actually give us the new three-dimensional structure of a liver made in a petri dish. Another way we now know a genetic damage mechanism that can lead to cancer is illustrated here. We're looking inside that chromosome, and you can see a strip of DNA sequences within that chromosome, and you can see they're in different colors so we can keep the order straight. Well, another mechanism of causing cancer is when sometimes the cell undergoes traumatic damage and that whole chromosome is broken into little fragments like that. Catastrophic chromosome breakage, the big name for the process is chromothripsis. What happens then, the normal cell mechanisms for DNA repair puts those fragments back together, but usually oftentimes in the wrong order, and some pieces are left out, and then the process we activate an oncogene or inactivate a tumor suppressor. So this process is another DNA damage mechanism that can lead to cancer. Now we're in the laboratory looking at lungs of a rat, a new mechanism of regenerative medicine for regenerating new lungs, and it starts with taking away the cells that are present and putting in new cells, decellularization, recellularization. We'll take a look at that this year also. Quickly, here's a sequence of events that shows the development of colon cancer, colon carcinoma, from normal epithelium lining the intestine on the left through a sequence of mutations to the cancer on the right, and we now know the entire sequence probably takes up to 30 years. And ladies and gentlemen, amazingly, experts are telling us if we could catch this in the first 27 of those 30 years, we could probably still avoid the cancer. That's a man-made rat kidney made in a laboratory. We're thinking in the future, people with kidney concerns, potentially fatal kidney problems, people on dialysis, we might indeed be able to make them a new kidney in a laboratory as part of tissue engineering, regenerative medicine technology. EMT is one of the most significant events involved in the lethality of cancer. Most cancer begins with an epithelial cell, but somewhere along the line those epithelial cells change, they morph to become mesenchymal stem cells, and that change is called epithelial mesenchymal transition, EMT. And why it's so important is once the cell makes that transition, then it can metastasize, leave the original site of the tumor, invade other tissues, get into the bloodstream, the lymphatic system, and spread through the body. So EMT is a necessary process for cells to metastasize in the development of a tumor over those 25 to 30 years. Here's a unique human body part that's been made in the laboratory. We have some women with a rare genetic syndrome where part of the female normal anatomy does not form properly. This is a man-made vagina, which can now be used for women with that rare syndrome. The last term I want to mention is stemness. Stemness, folks, is a term we use to describe the unique characteristics of a stem cell. There's four things that make up stemness. Number one, self-renewal. Stem cells can grow and make new copies of themselves. Number two, they can differentiate to make all the different parts of the body during differentiation. Number three, they've been through EMT, so they can migrate. They're migratory. They can move around the body. And number four, stem cells have the unique ability to avoid death caused by harmful chemicals like chemotherapy. They can actually pump the chemotherapy molecules out of the cell, or if there is damage, they can sometimes fix it. The last point I want to leave you with is this. All cancer begins with one cell. Well, what if that one cell that gets that first mutation that leads to cancer is a stem cell? Then we have a stem cell that's going to give rise to a tumor, but that stem cell has stemness. Those are called cancer stem cells, and that's a unique part of what we'll be talking about this year also. So those are a little overview, guys, of the program we're going to be doing around the country in colleges and high schools for 2014-15 school year. Please keep in mind we are available now on Twitter. If you want to follow me on my Twitter account, we're doing updates 2 or 3 times a week just on Twitter. Of course, you can always get in touch with me at samrhine.com. Any questions, comments, concerns, don't hesitate to do that. Want to come to one of our conferences, you can register at that website. Have any questions about anything we've talked about, give me a call or text me at area 317-439-7493. Hope to see you at one of our conferences this year. Thank you very much.